So we're very excited that the second PARP inhibitor for germline BRCA associated advanced breast cancer has now been approved by the U.S. Federal Drug Administration as of October 16th, when talazoparib was approved. And it's being evaluated by the European Medicines Agencies that's accepted it for review. So that's exciting because previously, for a long time we had no approved PARP inhibitors, which made no sense, and then we had olaparib. So now we'll have, as of this week, now have two PARP inhibitors that are approved in this setting. The EMBRACA trial actually provided the data that led to the approval. It was a registration study that randomized women with metastatic breast cancer who'd received up to 3 lines of prior chemotherapy for metastatic breast cancer and had a germline BRCA1 or 2 mutation to receive either talazoparib or treatment of physician's choice, which were standard chemotherapy agents. And again, it was a 2-to-1 randomization. So we actually, one of my colleagues presented data from a subset looking at triple-negative breast cancer, and similar to what we saw with the trial as a whole, progression-free survival was significantly prolonged, not only in the group as a whole, but also in the triple-negative subset. And it's really striking to look at the difference in response, which was dramatically higher with the PARP inhibitor than with the chemotherapy. But chemotherapy is generally cheaper than a new agent, so big consideration always is, is it really worth it? And why give it? Maybe you can just keep giving it later and later because chemotherapy is cheaper, and so why give the drug? The second thing that comes up is that all of these trials excluded patients who had disease resistant to platinum salts, because platinum chemotherapy resistance seems to have a similar mechanism than resistance to PARP inhibitors. So if you've already had disease progression on a platinum, your response rate and benefit from PARP inhibitors is not 0, but it's less than if you didn't have that situation. So people have said, well, maybe we should use a platinum because it's so much cheaper, and not use the PARP inhibitor because we haven't yet seen a survival benefit from those trials. But we presented looking at the triple-negative subset data for patient-reported outcomes, and then there's also my colleague, Dr. Ettel, also published the PRO data for the entire study in Annals of Oncology. And what's really fascinating is the time to definitive deterioration, clinical deterioration, is dramatically longer with the PARP inhibitor than it is with standard chemotherapy agents, including vinorelbine, eribulin, capecitabine, etc. And patients also had a decrease in many different symptoms associated with quality of life, functionality, and just symptoms associated with breast cancer using a breast cancer specific scale. So either you got a whole lot better in terms of quality of life and global quality of life and functionality, or you had much less deterioration in your symptoms. And then if you looked at time to deterioration on pain, it also was dramatically longer with the PARP inhibitor than with standard chemotherapy. So I think that when you take that data as a whole, and there actually was data on the hormone receptor positive subset as well, presented at the Young Women's Conference in Lugano in the last week, it really suggests that not only are you having a better response and longer progression-free survival with this drug, but also patients are living a better quality of life. Global quality of life, functionality, and symptoms are better for these patients than if they are receiving standard therapy. So I think it really provides a lot of important supportive data, which was highlighted at this meeting when we're evaluating new therapies, that is critical to have in order to make these choices.